I hope you all would be doing good. And today we are back with another important lecture. And for today's class, we will see that how come the other type of the white blood cells that we were referring to them as a lymphocyte, they would contribute a role into be saving your body against a pathogen once they have got into the body. And as we have said in the previous class, that these internal immune response is basically provided by two type of the white blood cells, like we have these phagocytes and then we have got these lymphocytes coming. So we were done with understanding that how the phagocytes they carry out the phagocytosis, and by that how come they would eliminate the pathogen out of the body. And now, today we will see that how come the lymphocytes they would provide the primary immune response and the secondary immune response that we have got in the body. But before I go, what you have to see is that these lymphocytes, by other mean, they are also called as the B lymphocytes or the B cells, and in them we would have the plasma cells coming and we would have the memory cells coming. But before I proceed with the detail of the primary and the secondary immune response, what you have to keep it in your mind is that these B cells they would give a specific immune response. Unlike the phagocytes that were attacking on any type of the pathogen which was getting an entry into the body, these B cells that we have got, they would be specific to be carrying the reaction, the immune response against one special type of the pathogen. So now you see, the idea is that prior to the entry of the pathogen, our body would make multiple copies of the lymphocytes. Like this is the same way we live in a country, and in the country we train the army, we train the air force and the other forces, special forces as well. And this is not the case that after the enemy has entered into our country and we start training the army personnel, and we do it prior to the entry of the enemy. So likewise, my body would be making multiple copies of the lymphocytes prior to the entry of the pathogen, and when they are being developed or when they are being produced in the body prior to the entry of the pathogen. So each of the B cell or the lymphocyte that we have, so they would develop a different shape of the protein receptor in their cell surface membrane. Like if you were to see this A lymphocyte with me, so this has got a different shape of the protein receptor. If we have got this B lymphocyte coming with me, so again you see compared to the A, the shape of its protein receptor is different. Likewise, for this last lymphocyte that I have drawn for you, so this again has a spatial structure of its protein receptor. So this means that this lymphocyte would only be able to bind with this pathogen or the antigen, and this B would go with this, and the C would go with this. C at any cost would never be, or the lymphocyte that I have shown you with C. Now here I must clear that ABC does not mean the type of lymphocyte. This is just different lymphocytes having different protein receptors. So the C at any cost would never bind with this. A at any cost lymphocyte would not bind with this. Only this lymphocyte whose protein receptor shape is complementary to the antigen that we have got on the surface of the protein receptor would bind with it. And this is what we mean by the term that the B lymphocyte they provide the specific immune response. So if I were to just read this for you, so what I'm going to say is that these immune response, by the word specific immune response, what we say is that prior to the entry of the pathogen, what my body would do that it would produce multiple copies of the lymphocytes or the B cells. And these B cells or the lymphocytes, now they have developed different protein receptors and that allow them to bind with only one specific or definite kind of a pathogen, not with all of them. And this binding of the lymphocytes with the antigens or with the pathogen would depend that what similarities or what situation they have got in terms of complementary shape. Only the lymphocyte whose protein receptor is complementary to the antigen present on the surface of the pathogen, they would bind with it and the rest would not bind with it. Now, after you are clear with this concept of the specific immune response, now here you see that our lymphocyte that we have in the body, so they give two type of the immune responses. And among the two type of the immune responses, the first immune response that they would carry out in our body, So this is entitled as, or we call this as primary immune response, we say. And the second immune response which these lymphocytes would provide, so this is entitled as, or we call this as a secondary immune response. This is the timing that actually do the difference. If the pathogen would enter for the first time, so the primary immune response would be initiated. And if the pathogen would enter for the second time, and if this is the same pathogen, so my body would carry out the secondary immune response. So we initially have to see that what this primary immune response actually is, or what is the mechanism of this primary immune response. So we have to see two way around. Here we have got the diagram on this side, and here we have got the description. So the first thing is that the pathogen, it would get an entry into your body. Okay, and how come this pathogen can get entry into your body by aerosols, by some insect vectors, by having some contaminated food or the contaminated water? So this antigen or the pathogen having an antigen on its surface, so this has entered into the body. Now, after it has entered into the body, so the B cells, like we have got multiple of the lymphocytes or the different kind of the B cells with us, and all of them, they have different shape of the protein receptor, but only that lymphocyte whose shape of the protein receptor is complementary to the antigen which is present on the surface of the pathogen, so it would bind with it. And after the lymphocyte has binded with a specific pathogen, so this selected lymphocyte would now undergo mitosis. Again, by mitosis, what we mean is that it has to increase the number of cells now. Like I would never want this lymphocyte to increase in number, but rather than this, because this is the lymphocyte that we have selected for now. Now, once this selected lymphocyte would undergo mitosis, so it would produce two subtype of the cells. Like we have this antibody secreting cells being produced, and these are the plasma cells. And at any cost, we have to be keeping the memory of this pathogen so that later in the life our body should be prepared. So not only the antibodies are released, but at the same time, my body would also be producing a memory cell, and that memory cell would keep a track of this pathogen. So later, when this pathogen would enter for the second time, so my body should start producing these antibody molecules rapidly. So if I were to read all of this, so what we would say that first the pathogen having an antigen on it, it would get an entry into the body, then the B cells whose protein receptor shape is complementary to the antigen, it would bind with it. After this has binded, so that selected lymphocyte would divide by mitosis and it would produce the plasma cells and it would produce the memory cells. Now, as of this point, we are not sure that how come the lymphocyte have eliminated the pathogen out of the body in the primary immune response. So we have to see that after the lymphocyte have produced these antibodies, so how come these antibodies would eliminate the pathogen out? So now the situation is that these antibodies, what they do is that they would clump, they would agglutinate, or they would stick the pathogens together. And when they would stick the antigens or the pathogens together, so this means that these pathogens would not be allowed to spread around in the body. The other thing is that these antibodies they create pores in the surface of the pathogen, and when these pores or when these holes would be generated within their cells, so the water would enter. And when the water would enter, so as a result of that, these pathogens they would burst and they would be killed. The other situation is that these antibodies that we have got, so they have an ability to also bind with the toxins. Like this is a pathogen. This pathogen is not itself going into the cell, but rather than this is producing a toxin out of it. And this toxin have an ability to harm my cell. So what the antibodies they would do is that they would neutralize these toxins by binding with them. Now the other idea is that these antibodies they have trapped this pathogen. Now it should be ingested or this should be eaten up by the phagocytes. So by this what we can understand is that these antibodies they also have an ability to enhance activity of the phagocytes. Like after these antibodies have catched the pathogen, now they would take it to the lymphocyte, and once this would be taken to the lymphocyte, now this would be ingested. And when this would be ingested, then finally my body would overcome the harm of the pathogen. So if I were to read these points, so what we say is that the lymphocytes, after they have divided by the mitosis, so they produce the plasma cells. These plasma cells, they have produced the antibodies. And these antibodies, what they did initially was that they clumped or they stick the pathogens together, so they cannot spread around. And these antibodies, they create the pores in the walls of the pathogen, so water would enter into it and it would burst them. The pathogens, they were producing the toxins, so the antibodies would bind with the toxins and would neutralize them. And the antibodies are also increasing the activity of the phagocyte. So this is what you have to remember is in terms of the primary immune response. Now what we have seen above is that these lymphocytes in the primary immune response, they have made these memory cells. And these memory cells, what they are doing is that they would keep the track of the pathogen, that what entered into the body for the first time. Now, if I say that later in life, the same pathogen has entered again, so what the body would do, or what the lymphocyte would do, that they would bring about the secondary immune response. Now, the idea is that this secondary immune response would be initiated or the started when the same pathogen or the antigen, when this would enter in my body for the second time. From the word secondary, the idea is second time. And as a result, the memory cells that we initially formed during the primary immune response, now they would bind to the pathogen's antigens and they would rapidly start producing the antibodies, and in that way, our body would overcome the pathogen quick. That is why the primary immune response that we have is slow, because it takes time to be making the antibodies and the memory cells. And the secondary immune response is fast because my body already has got the memory cells and they already know that what pathogen this is, and they would rapidly be producing the antibodies, and that would help to be overcoming this pathogen that has entered. So I hope you would be clear with that how come these lymphocytes they bring about the primary and the secondary immune response with an intention to be saving your body from the attack of pathogens.